Hi guys, welcome back to my channel. I am so glad to see you all here. So far in all my videos, I have basically spoken about transcriptomic data, that is RNA sequencing or microarray, more of RNA sequencing. And going forward, I want to talk about other types of data that we routinely use in bioinformatics, like for example, DNA sequencing, like whole genome sequencing or whole exome sequencing. So before jumping on to making videos using other data types, I wanted to make a video where I talk about different types of high throughput data. In this video, we will go over each of these commonly used data type and talk about what it captures, what questions it helps to answer, and what are some of the most commonly performed analysis. Let's say if we were trying to understand the mechanisms underlying a disease. So ideally what we want to do is we want to identify biomarkers that are responsible for disease development, progression, and that can help us to come up with treatment which can interfere with those mechanisms. So to further understand the role of these markers, the data is collected at various levels. So, coming back to the central dogma, we know that DNA is transcribed to RNA and RNA is translated into proteins. So, at each level, there is various types of data that are collected. So, at the DNA level, we have DNA sequencing data, which is whole exome sequencing, whole genome sequencing, and targeted sequencing. And I will be talking about the details of each of these data type in further detail in the upcoming slides. At the RNA level, we have microarray or RNA sequencing data, and we have spoken about this extensively in all our previous videos until now. And at the protein level, we have reverse phase protein array, mass spectrometry. So these are the methods that are used to quantify as well as classify and identify various proteins. In addition to these data types, there are also data that are captured that do not necessarily fall under the central dogma. And some of the commonly used data types are epigenetic data. So they capture epigenetic modifications, and there are various methods to capture these modifications. And these modifications influence the expression of genes. So we'll be talking about this in further detail in the upcoming slides. And lastly, we have metabolome, which measures the metabolites in the system. So metabolites like amino acids or lipids are measured, which can help us in understanding how the metabolite profile looks different in, let's say, a diseased patient versus a healthy patient. So we'll be talking about all of these data types in further detail, but the point here being that data is collected at various levels, and each type of data can be used to answer specific questions, and there are specific analysis that are performed which can help us leverage the information that is captured by each of these data types. So we'll start by talking about genome, and the data at this level aims to capture genes or genome level alterations. And it's important to identify these alterations and study whether it results in a dysfunctional protein. So the alterations in genes are single nucleotide polymorphisms, insertions and deletions, copy number variations, fusion genes, translocations, inversions. There are various kinds of alterations that can occur at the DNA level. And it's important for us to study this and identify which are the mutations or the alterations that are disease causing. So to give you an example, SNPs in genes like VCAM1 and ARF-GEF2 gene are found to be associated with sickle cell anemia. Similarly, an alteration like deletion in a CFTR gene is associated with a disease called cystic fibrosis, and alterations like copy number variants in the HLA gene is found to be associated with rheumatoid arthritis. So the most common high throughput sequencing data type in this category is whole genome sequencing or whole exome sequencing. The only difference between whole genome and whole exome is that the whole genome sequencing sequences the entire genome, that is all the genes in the genome, and whole exome sequences only the protein coding regions of the genome. So as we know that only 1 to 2% of the genome codes for proteins, that is these genes are transcribed into mRNA, and the mRNA results into a functional protein. So the whole exome sequences focuses only on those regions which gives rise to a functional protein. In addition to the whole genome and whole exome sequencing, we also have targeted sequencing, and this method isolates specific regions of DNA to analyze them for mutations. So this technique can identify variations in specific genes, which can be very useful if one only has to diagnose or rule out distinct variants. So for example, when screening for a particular genetic disease, so you only sequence specific regions in the genome, the regions of interest. DNA sequencing data is often used to answer questions like what are the variants, what are the disease causing mutations, where are the mutations in the genome located, and some of the commonly performed analysis is variant discovery, identification of which of the variants are disease causing, and performing genome assembly and annotation if it's a genome that hasn't been previously sequenced. Now, talking about epigenome and epigenetic changes, I want to give you an idea as to why it is important for us to understand the epigenetic changes and to capture these epigenetic modifications. So, in our eyes, we have specialized cells that turn on specific genes that make proteins that can detect light. In our blood, we have specialized cells that make proteins that can carry oxygen from the air to the rest of the body. Both these types of specialized cells consist of the same nucleus, basically the same DNA. The differences among the cells are determined by how and when the different sets of genes are turned on for various kinds of activities. Our body is made up of trillions of cells specialized for different functions in brain, in muscle, and in bone, and each of these cells carry essentially the same genome in the nucleus. So basically, the epigenetic changes control many of these changes in the gene expression, which results in the specialized set of functions each of these cells carry in different organs. So basically, epigenetic data captures the set of chemical modifications to the DNA and the DNA associated proteins in the cell, which results in altered gene expression, and these modifications are heritable. So some of the modifications that affect gene expression is methylation, that is addition of methyl groups, histone modifications, and binding of the transcription factors. A method called ChIP-Seq, that is chromatin immunoprecipitation assay, is a method that will provide us with the patterns of transcription factor binding sites to the DNA and patterns of histone modification. So this method helped identify the gene FOXA1 as having a prognostic role in facilitating estrogen receptor binding in estrogen receptor positive breast cancer. So before I go into the details of what are the types of epigenetic modifications that are captured, I want to talk about how DNA is organized in a cell. So DNA is this double helical structure which is wound around tightly around these proteins called histones. So there are 8 histones that form a structure like this, and around which DNA is tightly coiled. So the entire DNA is coiled around these proteins, and this structure is further coiled into a strand that is called chromatin. Now this chromatin is further coiled into forming a chromosome arm. So if you pick any cell, if you look at the nucleus, you will find many chromosomes. And if you were to pick a chromosome and extend an arm of the chromosome, you will find a chromatin, and if you zoom into the chromatin, you will be able to see that the DNA is tightly coiled around these protein structures. Now in order for a gene to be expressed, these structures has to be relaxed so that there is some amount of accessibility for the promoters and transcription factors to bind to DNA in order for transcription to happen. Epigenetic modifications to the histone or DNA can affect this structure. So basically, there are certain modifications that can cause the DNA to pack more densely around these histone proteins, and since they are packed so densely, there is no space or accessibility for the transcription factors to access the genes or the promoter regions of the genes, and since they cannot access these regions, the DNA is not transcribed and the gene is not expressed. But there are certain modifications which causes the structure to relax a bit, and when they relax, there is more accessibility of the transcription factors to bind to the promoter regions of the gene, and when they are allowed to bind, the genes can be expressed and ultimately resulting into the protein expression. So a lot of these expression of proteins depend upon the epigenetic changes which controls the expression of gene. Talking about modifications, one type of modification is DNA methylation, where methyl groups are added to the DNA. So methods like whole genome, bisulfite sequencing, methylation arrays can be used to quantify DNA methylation status. In addition to DNA methylation, we can also get the patterns of transcription factor binding sites and histone modifications using method called chromatin immunoprecipitation assay, which provides us with location and the intensity of the binding of these transcription factors, as well as information on the location of these histone modifications. So in addition, we can also get the sequences of the regions that are not bound by histones by performing chromatin accessibility assay. So one method that is commonly used, that is ATAC-Seq, is used to capture regions that are not bound by histones. So since these regions are not captured by histones, they are accessible by transcription factors to bind to the promoter regions, to the genes in that region, and hence the gene transcription can occur. So having that information can be useful as to understand which of the genes in a particular condition or in a particular state are free of histone, and basically they are accessible, and the genes in that region are being transcribed. Lastly, we can also get the conformation of the chromatin. So chromatin conformation assays are used to study the physical interaction between the genes and the distal regulatory elements, as well as proteins that causes such looping of the chromatin. So one common assay to study chromatin conformation is Hi-C. So epigenetic data can be used to answer questions like where does a transcription factor bind, which bases are methylated, where does the RNA binding proteins bind, and which parts of the genome are nucleosome free. And some of the commonly performed analysis are transcription factor binding site predictions, correlation of methylation status with the survival data, also correlating histone modifications with expression data. Now, coming to transcriptome, this is something that we have spoken and worked with extensively in many videos on my channel. So, basically, transcriptome examines RNA levels genome wide, both qualitatively and quantitatively, and it captures both the coding mRNAs and the non-coding RNAs. So mRNA is basically used to study drug response and molecular subtyping. The analysis of mRNA provides direct insight into cell and tissue specific gene expression features. Non-coding RNAs, on the other hand, play essential role in physiological processes, and dysregulation of non-coding RNA, that is a long non-coding RNA, has been implicated in various diseases like myocardial infarction, diabetes, and cancer. RNA expression patterns were also used to define four breast cancer subtypes which were associated with patient response to chemotherapy. Other types of non-coding RNA like microRNAs have been associated with relapsing remitting multiple sclerosis and dormancy of the HIV in patients treated with antiretroviral therapy. So there are two methods to study the transcriptome, that is microarray and RNA sequencing, and we have previously used both these type of data in our analysis. We have also extensively spoken about the differences between both these methods in the previous video, so I'm not going into the details of them. So just quickly going over these methods, the RNA is extracted from the sample and is reverse transcribed to complementary DNA. In terms of RNA, in terms of microarray, the complementary DNA is labeled with fluorescent tags and is allowed to hybridize with a complementary probe that is attached to an array. And the ones that have successful hybridization will result into higher fluorescent intensity, and these fluorescent intensities are picked up by the fluorescent scanners. So in order to use this method, one requires a prior knowledge of the sequence that one wants to capture, since the probes that are attached to the array are complementary probes to the sequence of interest. In terms of RNA sequencing, the complementary DNA is fragmented and is fed into a sequencer. The sequencer gives out reads, and these reads are computationally aligned to a reference sequence. The amount or the number of the reads that are aligning to particular regions are quantified, and that's how we get the gene quantification or isoform quantifications. I have previously demonstrated how can one align these reads to a reference genome and can get quantifications using RNA sequencing data in one of my previous videos. So if you're interested in knowing how to perform that analysis, I will make sure to link that video somewhere in this video or put it in the description section below. In order to use RNA sequencing, one does not require to have prior knowledge of the sequences, and it allows the detection of known as well as novel transcripts and isoforms. So transcriptomic data can be used to answer questions like which genes, isoforms, or microRNAs are expressed and how much are they expressed. Is there a differential expression between disease state or healthy state? And some of the common analysis performed using transcriptomic data are a quantification of mRNA and isoform expression, differential expression analysis, or finding genes that are functionally associated by finding genes that are highly correlated by performing weighted correlation network analysis. Now, talking about proteome, at this level, we study the abundance, modification, and interaction of proteins with other proteins. So, basically, proteomics helps us in understanding how expression of proteins, their structure and function can cause the diseases. These approaches allow us to compare the protein profiles of cells in healthy versus diseased individuals that can be used to identify proteins associated with disease development and progression. Methods like mass spectrometry or liquid chromatography mass spectrometry can be used to study the sequence and structure of proteins, each protein having a unique weight, that is the fingerprint, that can be used to identify their presence in a sample. Other methods like an array based technology is also used, and in one of the cases this method was used to identify protein targets which were for a tailored treatment for a patient with inflammatory disease of the retina, which resulted in reversing the sight loss. The functions of a large fraction of protein are mediated by post-translation modifications such as proteolysis, glycolization, phosphorylation, and ubiquitination. Such modifications play key roles in intracellular signaling, control of enzyme activity, protein turnover and transport, and maintaining overall cell structure. Methods like mass spectrometry can be used to directly measure such covalent modifications by defining the corresponding shift in the mass of the protein. Methods like chromatography, protein microarray, mass spectrometry are frequently employed methods to detect biomarkers or proteins of interest for diseases and detecting therapeutic targets. So, the data from these methods can be used to answer questions like what proteins do the protein of interest interact, how much is the protein expressed, and whether specific mutation affect the protein structure. And some of the commonly performed analysis is to identify or quantify a protein, performing or building a protein-protein interaction network, and generating 3D structure of the protein to understand protein function. After proteome, now we want to talk about metabolome, and metabolomics is the study of metabolites produced during biochemical reactions. Like proteins, metabolites are identified and studied using methods like mass spectrometry, chromatography, and nuclear magnetic resonance spectrometry, which helps in generating metabolite profiles. Now, metabolite levels and the relative ratios reflect the metabolic function, and out of normal ranges are indicative of disease. And this is important in drug discovery, which helps us in understanding the metabolism of a drug and its potential associated toxicities. Lipidomics, which is a subfield in metabolomics, is the study of lipid levels, such as cholesterol and triglycerides in blood and tissues. And one study characterized a lipotype in individuals that corresponded to a potential risk of developing metabolic syndrome. Another study discovered that in post-surgery patients with recurred epithelial ovarian cancer, it was found that these patients had high level of lipid and amino acid metabolism. So, quantifying the amount of metabolites in deceased patient versus a healthy patient can give us an idea on the metabolic profiles of these and can also give us further clues on the underlying mechanisms. Metabolomics data can be used to answer questions like what are the toxicities associated with drug metabolism, what is the metabolic profile of a normal versus a diseased individual, and some commonly performed analysis here would be to quantify small molecule abundances. Now, completely switching gears in terms of context, because so far whatever we have been discussing was in context of eukaryotic organism, but metagenome is something that has gained a lot of momentum in the recent years and is extremely useful in the study of prokaryotic organism. So, metagenome is the recovery and complete sequencing of genetic material extracted directly from environmental samples, and this process of creating a metagenome is referred to as metagenomics. Metagenomics particularly offers the advantage of analyzing genomes of non-culturable microorganisms such as viruses. So, traditionally, microbiologists used to apply cultural techniques and microscopy to identify and characterize microorganisms. These techniques are time consuming and provide a limited vision of a microbial community. But metagenomics applies a suite of sequencing technology and bioinformatics tools to directly access the genetic content of the entire communities of microorganisms. It also allows detection of rare bacteria or microorganisms that cannot be cultivated or cultured in the lab. It also enables to study a population diversity within a sample and allows to track changes in the population in response to a treatment or environmental conditions. Metagenomic sequencing involves targeted metagenomics. This method targets a specific region of a genome, that is 16S rRNA or 18S rRNA, that is shared across multiple organisms and samples. It also involves shotgun metagenomics, which basically sequence everything in the sample. Data from metagenomic sequencing can be used to answer questions like what organisms are present in the sample, do different populations interact, what genes are responsible to confer resistance to antibiotics or environmental conditions in an organism. And some of the commonly performed analysis are marker gene analysis, performing subtype or clustering analysis, performing genome assembly, gene prediction, and functional annotation for genomes that have been not previously been sequenced. So that's all I had for today's video. This was a quick simplified walkthrough of types of high throughput data. I hope you found this video helpful and informative. If you did, please consider hitting the like button, subscribe button, and share it to as many as you can. And please feel free to leave your comments under the comment section. Until next time, see you.